Okay, my Biology 107 fans, I'm just going to recap where we ended up at the end of our last lecture. We were talking about RNA and can it act as a catalyst, right? So can RNA act as a catalyst? And we now know that that is possible due to the experiment that Altman and Czech did. And I'm going to go over that just so that you remember what it is. They were looking at this free-living ciliate, a protozoa that is found in water, Tetrahymena, and they found that there are RNA molecules that are able to catalyze the reaction that adds nucleotides onto existing nucleotide sequences. And that experiment shown here is, remember, this is where they took a long string of nucleotides, RNA, and let's just say, they did this in 1 million test tubes. They took 1 million long strings of RNA, and into that test tube that had that one long strand of RNA, they added in a small template piece of RNA, so this is the template. And they added a primer, and they let the reaction go for a certain amount of time. And what they were looking for was, can additional nucleotides be added onto this string? And when I say can they be added on, I'm talking about, can this large RNA, shown at the top, catalyze the reaction? Can it act as an enzyme to make a phosphodiester bond? So each nucleotide comes in, it base pairs appropriately, and the link between the nucleotide on the end of the primer and the new incoming nucleotide is a condensation reaction that is catalyzed by this large RNA. And cutting to the chase, we know that that's exactly what happened. This image of gel electrophoresis shows that at time 0, we have this single band, and that's just the primer, and that over time you get these larger and larger bands showing up because the RNA in the test tube helps add nucleotides on over time. So at 24 hours you see lots and lots of bands on a gel, and that's because the RNA acted as an enzyme. And it has a very special name. It's called a ribozyme because it's a RNA molecule that acts as that enzyme. Ribozyme. So this provided the evidence that RNA could have been that first self-replicating hereditary material enclosed in a membrane that was the first life form. Yay! This really is why we think RNA was that first life form. So hopefully that brings us to sort of the end of looking at these macromolecules. We've looked at RNA, we've looked at proteins, talked a little bit about carbohydrates, but we'll talk more, I think. And then what I'd like to switch to now is, what does having this genetic material mean? So what is the genetic code? Okay, very, very important concept, and I'm not saying very, very so that you just don't pay attention. I'm saying it because if you don't know this, you can't get out of 107. You can't pass. So there is something called central dogma. The central dogma, dogma is something that is believed, and it's not just sort of believed. This is like a rule, something hard and fast that we absolutely think is the only way that it could be. So the central dogma of biology is that DNA is transcribed, so this is the process, into RNA, and RNA is translated into protein. And this is absolutely something that you must know. DNA is transcribed into RNA, and RNAs are translated into proteins. So DNA is the hereditary material that allows you to make all of the proteins that your body uses to make up cells, tissues, organs, everything that makes you who you are. Now, when we start talking about DNA and its hereditary value, okay, there's a couple terms that will get thrown around, especially when we start talking about Mendel. So we'll talk about genotype versus phenotype. Genotype are the actual genes that you have for a particular trait, and the phenotype is actually what you end up looking like. And so we are organisms that have 2 copies of every gene. We are diploid. Diploid. Di meaning 2. And you have 2 of every gene because you have 1 chromosome from mom and 1 chromosome from dad. So every single chromosome that you have 2 of them. So you have chromosome number 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22. And then your 23rd pair are your sex chromosomes. But you have 2 number 1s, you have 2 number 2s, you have 2 number 3s, etc., etc., etc. And for sex chromosomes, you either have XX or XY. Now there are some odd circumstances where that's not exactly true. Okay, there are some XXY and XYY individuals. All right, but in general, you have 23 pairs of chromosomes. And that means you are diploid for every single gene, meaning you have 2 of every single gene. Okay. Now, interestingly, a lot of people recognized early on that if you somehow mutate a gene, you can mutate that gene and then look at an organism and say, what happens to the organism when you mutate that gene? We call those knockouts. So that's if you actually mutate a gene and make it completely defective. Null or loss of function mutants mean either nothing happens or you lose the function of that particular gene. And so there's lots of ways that we can begin to understand genotype and phenotype. So here it is again. DNA, and this arrow here that I'm going to make in red, the word that you want is DNA is transcribed, or DNA by the process of transcription is made into RNA, and then RNA by the second arrow is translated into protein. And what are proteins made out of? Proteins are made out of amino acids. So what is RNA made out of? RNA is made out of nucleotides. So nucleotides get turned into, right, a sequence of nucleotides gets turned into an amino acid sequence. And so what we're saying here is that indirectly, right, through RNA, DNA codes for proteins. In eukaryotic cells, this image shows you that DNA is in the nucleus and it gets transcribed into the nucleus into an mRNA. That mRNA exits the nucleus, and in the cytoplasm it meets up with ribosomes where it gets translated into a protein. Some of those proteins go right back into the nucleus and act in the nucleus. Others of them can be exported out of the cell or taken to the ribosome, on the ribosome to the ER, where it gets processed and sent through the ER ergic Golgi, the exocytic pathway, on its way to its final destination. So this central dogma, really, really important. All right, here are the words, right? DNA goes to RNA. That's called transcription. Transcription needs something that can make a string of RNA. That's called a polymerase, because you're taking monomers, putting them together into a string of polymers. Okay, so we call this an RNA polymerase, and transcription uses a sequence of DNA, right? So if there's a sequence of DNA, remember I told you I could draw it like this, it still means that it has bases on it. And what has to happen is you have this as a template. You're going to see that you actually need a little primer to do transcription, and that allows for us to then add on here, and you add always going in the 3' direction. So you have a template with a primer, and we can add nucleotides using this polymerase. Interestingly, this polymerase is a protein. Even though it's an RNA polymerase, it's because it's making RNA. Okay, it's not a ribozyme. And this is done because of complementary base pairing. We know what to add on to the new strand because we're complementary base pairing to the template strand. Okay, so template, you need to know these terms. You need to know what a primer is. It's a shorter piece of DNA or RNA that allows for us to add on to it. Okay. And complementary base pairing. Now RNA in central dogma is then translated in a process called translation into a protein, and that uses ribosomes. And interestingly enough, okay, ribosomes have ribozyme activity, and we'll talk more about this later, but just start trying to understand this in your head. Now this is where it gets complicated. All right. So the genetic code, how is it possible? So maybe let me pose this question to you. Let's slide this up here. And the question that you have to think is, how can a sequence of nucleotides end up coding for a protein? You know what the nucleotides are. So you have, in DNA, you have 4 nucleotides, A, C, G, and T. And in proteins, what do we know we have for proteins? There's 20 amino acids. So how can nucleotides, any individual nucleotide, encode for an amino acid? If each nucleotide, right, if you said A coded for 1 amino acid, okay, so that would give you 1, and C coded for 1 more amino acid, that'd give you 2, G encodes for 1 more, that gives you 3, T. Well, if each nucleotide encoded for an amino acid, that would only give you 4 amino acids, so that can't work, because we know we need to at least make 20. And that's where these images, I'm going to zoom back in. Okay, in this image, what the point is, okay, clearly here, 4 nucleotides that's not going to work. But if you take 2 nucleotides together, right, if you pair 2 nucleotides, and so you have every combination of the 2 nucleotides, you have U paired with a U, U paired with a C, U paired with an A, U paired with a G, a C with a U, C with a C, C. Okay, how do you get this number? It's 4 nucleotides squared. This is 4 nucleotides to the first. So 4 squared equals 16. Still not enough to encode for the 20 amino acids that we need. And so somebody came up with the idea, let's try 3. If you do the math, it's 4 to the 3rd, so 4 times 4 is 16, and 4 is 64. Well, 64, definitely can cover 20 amino acids, and it turns out that this is exactly what happens. So each triplet is called a codon, as in it's coding for an amino acid. And so we know that the genetic code allows for 64 combinations of 3 nucleotides. So why do we need 64 if you only have 20 amino acids? Well, there's something called redundancy. So more than one codon can code for the same amino acid. And where we end up with this chart, okay, I'm going to scrunch it down just a little bit to get it all on the screen. Okay, and so if you look at each of these codons, so here they're showing, this is a codon table. On the left-hand side is the first base in the codon table. So let's say you're looking at a U, and then the second nucleotide in the sequence is a C, and then the third nucleotide, so that second one is on the top, and then the third nucleotide is off to the side here, and let's say it's another C, so we go across to UCC. Well, that encodes for a serine. Okay. Serine is a amino acid. It has a 3-letter symbol called SER, and it has S as its 1-letter symbol. And so every different codon, right, you can look at and say, what does that encode for? Now notice, all 4 of these codons encode for serine. And if I go back and erase some of this, you'll be able to see since I covered it up. But, so this means UCU, UCC, UCA, and UCG all code for the same amino acid. Interestingly, over here are 2 more. So this means serine can be encoded in the DNA or RNA by 6 different codons. And so that is an example of redundancy. Now there are just a handful of sequences that are used only once. So one codon that is only used for one amino acid is methionine. Now, if we look at this chart, I hope you notice that there's U's. So what does that mean? Are we looking at RNA or DNA? You should immediately say, if it has a U, we have to be talking about RNA. And how does that work, right? We were talking DNA indirectly codes. So DNA indirectly codes for proteins, right? Because DNA gets transcribed into RNA, and then it is the RNA that gets translated into the protein. So ultimately, DNA is what is coding for the protein indirectly through this RNA. So AUG is the single codon that is the start codon. And it's not just the start codon, it encodes for a particular amino acid called methionine, MET. It's the only one that has a single codon. All the rest have multiple codons. So that's the start codon of any protein that's going to be made, is going to be AUG in the RNA. Okay. So here is another example of that codon table. Okay. You need to be able to read them no matter which table I give you, and I'm not going to give you, so I'm actually going to not give you first, second, and third. If you can't remember that you start on the left is your first one, on the top is the second one, and on the right is your third one, that's not going to be my problem, because sometimes you get this codon chart and it doesn't have all the information on it. All right, so let's say you have UAU. That encodes for an amino acid tyrosine. Okay? We'll do some practice of these in class. So here's the chart that I'll give you. It's very simple. Okay, so you know that if you have a sequence of RNA that you're going to be looking for the first nucleotide in the codon here, the second nucleotide on the top, and the third nucleotide on the right-hand side. So here's a typical little example I might give you. And this one actually is too simple because I haven't done everything in this that actually happens in a real RNA sequence. But here's the sequence. AUG, which I just told you, encoded for methionine. Okay, and what you want to do is you look at every 3 nucleotides, and sometimes it's good just to put a line right in between them so that you can see that you're actually counting 3 at a time. Now in this sequence that I've given you here, right, if you go back and look at the chart, what it means is that you have methionine, aspartic acid, leucine, glycine, arginine, aspartic acid, leucine, tyrosine. I told you don't have to be able to draw the amino acids, but you have to be able to recognize their names. These are the names of the amino acids based on the codons in this particular RNA sequence. How do I know this is an RNA sequence? Because it has U's in it. Okay? Now, I'm just going to add one more slide here. Throw in one of the monkey wrenches, okay, that makes students have some trouble. When DNA is transcribed into RNA, it isn't transcribed starting at the start codon. And so what happens is when DNA is transcribed into RNA, what you end up with is sequence ahead of that first AUG. Okay, and then this sequence continues until there's a stop codon. So let's see, I think I need a UAG is a stop codon. And then the DNA doesn't stop there. It keeps going until it gets to the end. And there are regions of nucleotides, RNA nucleotides, that are called the 5' UTR. And UTR stands for untranslated region. So there are nucleotides ahead of the first AUG, and then there are nucleotides downstream from the last nucleotide that encodes the stop. And this sequence here is called the, right, so even the UAG is part of this, is the 3' UTR. So if I were to give you untranslated region, if I were to give you a sequence of nucleotides that I asked you to take a look at, okay, right here I give you a sequence of nucleotides from the 5' to the 3' region. What you really want to do is you want to scan along, so you're going to scan along, and everywhere there's an A, right? Let's say there's an A there, you're going to say, is it an AUG? So you have to look for the first AUG. Okay, if it's not an AUG, you keep going. Here's a C, an AG, and no. Okay, here, A, no. And then you come to this one and you say A, UG, and so from here, what you do is you say, okay, here's my coding sequence. And that coding sequence goes until you reach a stop codon. Okay, and there are 3 stop codons. UAG, UAA, and UGA. So there are 3 stop codons, and wherever that stop codon is, so let's say you have the UAG, from there to the end, that's going to be your 3' UTR. And on this 5' end, this is going to be your 5' UTR. So that's how you're going to begin to look at these sequences of RNA to determine where is my gene. Okay, so the gene that was transcribed into RNA is now here, and right where the blue is, and that's what you're going to translate into protein based on the codons in the RNA sequence. All right, we'll work on this in class. Have a great rest of your weekend. Talk to you soon. Bye.